Bacterial capsules. So the learning outcomes of this session is that capsules being the outermost layer of bacteria are protective in nature. We will be able to comprehend the basic composition of capsules, the morphological and functional attributes of bacteria due to the presence of capsules, virulence of the bacteria which is increased due to the presence of capsules. Now let us just look at capsules as an overview with respect to structure and its functions. So when you understand or look at capsules in a bacteria, what has been observed is that capsules form the outer layer even beyond the layer present in gram-negative bacteria. So first and foremost, capsules can be present both in gram-positive as well as in gram-negative bacteria. And the gram-negative has an outer lipopolysaccharide layer. But even over and above the outer layer or the outer membrane of the gram-negative bacteria, you may have a capsule present. So these capsules, the consistency of these capsules can range from being semi-rigid to slimy. And also what is observed is that many bacteria can come together and together the capsules can merge with each other to form what is called as a biofilm. There is an interesting fact when one talks about sliminess. So this has been observed that when you have the water content in the capsules greater, it has been observed that the bacteria shows what is called as a mucoidal consistency, and when it grows in a petri plate, what's observed is that there are chances that you have the bacterial colonies formed in such a way that it seems as if they are flowing from the point of origin. So therefore what is observed is that the slimy portion of the capsule enables it to take up a certain morphology. So the outer envelope, although it may be semi-rigid or it may be slimy, it is well organized. So there is a structure to the capsule and it cannot be easily washed away as thought so. Now, these capsules generally are comprised of different types of polysaccharides. Interestingly, one is not observing that the polysaccharides are only made up of a single monosaccharides, but in several bacteria, one has heteropolysaccharides that form the capsules. And there are also, other than polysaccharides, that is carbohydrate moieties, one also sees peptide moieties present in the capsules. And definitely the capsules are associated with virulence, adherence, protection, and barrier. Now we will look at all of these in a little more detail. So when one looks at composition of capsules, what is interestingly observed is that capsules are generally repeating polysaccharide units, and these repeating polysaccharide units are heteropolysaccharides, with of course a few exceptions. So these exceptions basically are that you can have capsules made up of only peptides, okay? So you have different peptides coming together through different linkages to form a capsule, or you could have glycans and peptide coming in together to combine to form what is called as the capsules. So it's not necessary that a capsule is only a polysaccharide component. It can also have peptides present within its structure. Heteropolysaccharides per se is an understanding that the polymer that is formed is made up of, could be made up of glucose, rhamnose, mannose, N-acetyl glucosamine, manuronic acid, to a certain extent that one also observes in certain bacteria, the capsules having acetic acid. So therefore, there is a range of different monosaccharides that can be present in the capsules. Now, because they are sugars, what is observed is that when bacteria grows in a sugar-rich media, then it tends to form capsules. So if you leave it in a media containing more sugar, then bacteria that have the genes to produce the capsules are capable of forming the capsules around it. Uh, Another important factor that has been observed with heteropolysaccharides is that several of these monosaccharides can come together to form an organized cluster. and these have implications in how it is able to elicit an immune response or resist an immune response. Because one of the factors that is observed is that the presence of capsules in bacteria helps it to protect itself from the host immune system. That is one of the major functions of capsules. And therefore, such organized clusters that are present in the capsules are referred to as points against which you can have antibodies being formed. There is one other factor that is observed is that because they are polysaccharide units and most are sugars have the ability to hold water, and since they are able to hold water they remain hydrated. And also because there are several acid sugars that are present in the capsules, there are many capsules which are or can be acidic. The genes that are responsible for capsule biosynthesis are observed to be clustered in a certain part of the genome. So this is also very interesting. Most of the genes that form the capsules are present almost together as clusters, and it has a significance in the fact that when the environment calls for capsule formation, then the bacteria may be able to form capsules. So this is another thing that is reflected over here. So when bacteria grows in a sugar-rich media, it tends to form capsules, which means all the genes that are responsible for forming the capsules get activated, and they are present in close proximity to each other. So their regulation may be possible because of their close proximity. So these are just a comparative of the different compositions that can be present in different bacteria. So if you look at E. coli K30, you will find that uh, it has galactose, it has mannose, it has glucosamine. If you look at S. aureus, you will find there are N-acetyl manosamines. Then you have N-acetyl fucosamine. So you can see that how the S. aureus capsule is very different from that of the E. coli capsule. So is the case with Streptococcus pneumoniae, where you can see rhamnose as one of the monosaccharides present. Then you can also have S pyogenes. S pyogenes have what is again N-acetyl glucosamine present. And so the composition of the capsules in different bacteria can vary depending on what are the genes that are present in these bacteria that that enable the formation of particular polysaccharide constituents. Interestingly, Bacillus anthracis does not have polysaccharides in its capsule composition. It is mainly polyglutamic acid. So you can see there are repeating units of polyglutamic acids in its composition. So this is definitely an exception. As you can see, most of the bacteria have polysaccharides in its capsule, while you can see Bacillus anthracis has polyglutamic acid. So this is forming what is peptide, capsules that are formed. Now, what is also important is whether one can identify bacteria that form capsules. And in fact, what is observed is a lot of morphological changes have happened because of the presence of capsule. Now, one of the most highlighted experiment that also happens to be one of the experiments that talked about transformation in bacteria. So it has to do with what is the principle or what is the biological molecule that is responsible for transformation. And when Dr. S.F. Griffith was carrying out his experiments, he was basically trying to look to producing vaccines. Now, when he isolated these Streptococcus pneumoniae, they're more called as Diplococcus pneumoniae because they are present as Diplococcus. Now, these Diplococcus pneumoniae, when they were grown in blood agar plates, it was observed that there are two kinds of colonies that were formed. So very evidently, there are one group of Streptococci pneumoniae which are having smooth margin, as you can observe, very uniform, smooth margin, and these were observed to be smooth margin because they were encapsulated. So they had a capsule formed around them. Whereas the other colonies that were present are also Streptococcus pneumoniae, but these colonies were not smooth, instead the margin was rough. And it was observed that these rough colonies did not have capsule around it. So therefore, SF Griffith named the ones that had a smooth margin as smooth strains of Diplococcus pneumoniae, and the ones that did not have the capsule and formed a rough margin were called as the rough strain of Diplococcus pneumoniae. What was also interesting is that those which were encapsulated were virulent strains, and those that did not have the capsule were not virulent strains. So effectively what was observed is that when the smooth strains of Diplococcus pneumoniae were injected in mice, the mice suffered from pneumonia, and also many of them died. So therefore, it was observed that these smooth strains were quite virulent. They had increased pathogenicity and were able to go to an extent where the mice could die. While those with rough, while those that were rough strains, and when they were injected into mice, the mice did get slight or mild symptoms of pneumonia, but it did not prove to be fatal for the mice. And so the observation that the presence of a capsule enabled the Diplococcus pneumoniae to be more virulent than those strains that did not have a capsule around it. So effectively kind of understanding that those strains which were capsulated were able to protect themselves from the mice immune system, and therefore survived in the mice for a longer period of time. And because they were able to survive in the mice for a longer period of time, they were able to cause harm to the mice, which eventually led to the death of the mice. While those that did not have the capsule, they were not able to resist the immune defense or immune system of the mice, and the immune system of the mice were able to get rid of the rough strains faster. And therefore, although the mice did get some mild symptoms, they never suffered from pneumonia bad, and they were also not dying of the disease. So you can observe very clearly over here that the morphology of the bacteria gave a colony characteristic that enabled one to identify whether the bacteria had or is having a capsule or not. This is one interesting facet that is there. What is also observed that when one grows an enriched media, and one finds that there are certain bacteria that have a mucoidal appearance, they are not they have a very slimy kind of shiny slimy kind of consistency. So they are considered to be containing capsules around around them. So this is something that is very clear. So identification, the capsules present help in identification of a lot of bacteria because of the kind of colony characteristics that are there. The other way is to actually, to identify whether a bacteria has capsule or not is by carrying out capsule staining. Now, what is very clearly understood is that these capsules are not able to retain any stain, or they do not get stained by any dye. So they do not take up any color from any stain. But what is observed is the bacterial cell which is present inside the capsule can take up a specific stain, while the background can also take up a specific stain. So this is a typical negative staining method where the background is stained but the cell per se is not stained. Well, this is a specialized staining technique which ensures one to enable that what is a halo portion around the bacteria is basically not stained, and therefore it is the capsule, while the bacteria itself is stained. So therefore this is a distinctive way, a distinct staining methodology that is used to kind of know whether a bacteria has capsule around it or not. Now let us look at the major functions of capsules. By way of functions, one of the major functions, of course, is that the presence of capsules in a bacteria contributes to it becoming virulent. This is definitely observed. And many of the other functions that it has kind of kind of basically helps the bacteria to become more and more virulent. Now, one of the functions that capsule has is that of addition. Say, for example, you have a bacteria that has a capsule that has hyaluronic acid. Now, hyaluronic acid, when present as a part of the capsule constituent, say for example, is able to bind to a specific receptor on pharyngeal tissues. So therefore, there are chances that you would have the bacteria with the hyaluronic acid that is present in its capsule, enabling it to bind to specifically the pharyngeal region, and therefore the infection will be caused more at the pharyngeal level, forming what is called as pharyngitis. So say for example, you have group A streptococci. So group A streptococci is able to specifically go and attach to the pharyngeal system. So therefore, the addition, the binding of the capsule to the tissue can be because of the constituents present in the capsule per se. Second important function that the bacteria gains is that it is able to colonize with other bacteria to form a biofilm. Now, this also is extremely important because when they are present in the form of a biofilm, they're able to resist the attack of the host immune system even further. So therefore, its pathogenicity increases by 100 folds when it becomes colonized, or when it colonizes with itself, like it can be a single microbe colony colonization, or it can also be a polymicrobe colonization. So you can have biofilm formation because of the capsules present. Of course, it determines the morphology of the bacteria, and because the morphology changes, it is able to protect itself from various environmental surroundings that is present. Because it is highly hydrated, it prevents the bacteria from getting desiccated. So this is very, very important again. So again, what one observes is that the presence of capsules helps the bacteria to survive or sustain under very stressful conditions or different environmental conditions. That is something that is there. So these are the basic or major functions of the capsules. Now, one thing that we will definitely go further into is the virulence itself, because most bacteria that have capsules are associated with virulence. So the important facts that have been studied so far and have been attributed to the presence of capsule around the bacteria is that because capsules are present, the bacteria are able to evade the innate immune system of the host. So for example, there is something called as the complement system in the host immune as part of the host immune system, and these complement pathway is not able to act on the bacteria because of the presence of the capsules. One has already looked at is that because of the presence of the capsules, the bacteria can adhere to target cells. They are able to move across because of the kind of layer that it has, and it not necessarily being a very rigid structure. It is able to pass through different membranes or endothelium. So it is also able to, for example, allow bacteria to cross the brain barrier, okay, the blood brain barrier. So this is how effective the presence of the capsule can be for the bacteria. It also, okay, one interesting fact is that many of the polysaccharides that are present as capsules in the bacteria, the polysaccharides present in the capsules are very similar to host polysaccharides, and therefore what can also happen is that the bacteria may not be recognized as a foreign particle. Instead, because it has very similar polysaccharide as in the host, these bacteria are considered as self, and the immune system does nothing to remove them from the host, and therefore these bacteria will survive in the host, and because of that they're able to cause the virulence, or they are becoming more pathogenic. One another study has showed that because of the presence of capsules, the bacteria are highly resistant to phagocytosis. So phagocytosis is one of the means by which bacteria, other germs are removed from the host. But if The presence of capsule makes it resistant to phagocytosis. Already what it does is it has found out a means of evading the host immune system. And therefore, all these various studies that have been carried out show how just the mere presence of a capsule is responsible for making the bacteria highly virulent. So let us conclude. Capsules are made up of polysaccharides that vary from species to species, with a few bacteria having capsules made up of peptides or peptide glycan composition. The presence of capsules is responsible for specific colony characteristics, with many being mucoid or slimy or semi-rigid in its consistency. The capsules are responsible for multiple functions such as colonization, adhesion, transmission, stress sustenance, etc. Many bacteria have enhanced pathogenicity due to capsules contributing to virulence and protection from the host immune system. Thus, the study of capsule synthesis is helping in making drugs that can affect the encapsulation process, thereby decreasing the pathogenicity of bacteria. Please read this line very carefully. You would understand what is being said.